And so because you know that ribosomes are partially made of RNA, then you might deduce that there are actually nucleotide sequences that are kind of embedded within the ribosome, right? Because the ribosome is made partially of RNA, and here's the RNA sequence. And so we'll show you why this is significant in a second, that ribosomes are made partially of RNA. All right, there's other stuff that's needed in order to make a protein. So just like making a cookie, you can't make cookies if there are no ingredients. And so in order to make a protein, you need the ingredients. And the ingredients needed to make a protein are amino acids. So here's one example of an amino acid called methionine. And again, the function of amino acids during translation is that it serves as the ingredients. In other words, it's what proteins are made of. That's what you string together in order to make a protein. We mentioned that when you're making cookies, you need somebody to go to the grocery store and get the ingredients and then bring them back to the kitchen so that they can be used. Same thing is needed when a cell is making a protein. So somebody or some molecule needs to grab the ingredients and bring them to the kitchen so that they can be used. The molecule that does that during translation is represented by this thing. It's called a transfer RNA. So again, it's like the grocery store runner. It's the thing that grabs the ingredients or the amino acids and then brings them to the kitchen or the ribosome. One thing to note about transfer RNA, so this is yet another type of RNA. So we've mentioned that there is messenger RNA, right? And we've shown you the job of messenger RNA here. There's ribosomal RNA, which is the type of RNA that's found in ribosomes. And now we're saying that there is a third type of RNA called transfer RNA. And in a second, we'll show you why it makes sense that these things are called transfer RNA. But for now, what we'll mention is this. So if you remember during the first week of the quarter, we said that RNA can sometimes fold up upon itself in order to form secondary structures. And so here's proof of that. So transfer RNA, basically it's a single-stranded RNA that has become folded upon itself. Here's another picture of a transfer RNA to kind of hammer home that point. It's a single-stranded RNA, but it's kind of found some complementary sequences within the same strand so that they can form hydrogen bonds. And because those hydrogen bonds are forming, it gives transfer RNA kind of a T-looking shape. Right? So that's its secondary structure. And so because transfer RNA is a type of RNA, it has RNA sequences on it. And so that's what this is showing. We'll show you in a second why this is significant, that transfer RNA is made of RNA. Other things that are needed in order to make a protein, there's a couple of more players here, and here's what they do. We mentioned that when you're talking about making cookies, the person going to the grocery store to get the ingredients, it would help if you feed that person. Otherwise he'd get tired and he'd stop going to the grocery store for you. So you need food for the grocery store runner to give him energy. The thing in the cell that serves as the energy molecule is ATP. Right? So the role of ATP during translation is that it provides energy for the transfer RNA to go grab the amino acids. And then lastly, we mentioned that when you're baking cookies, it would help to have a timer to let you know when to take the cookies out of the oven. Same thing goes when you're making a protein. It'll help to know, or it'll help to have something to help end the process. And that's what this thing represents. It's called the release factor. And it's kind of like the timer, right? It helps the process end. So here are all the things that are needed in order to make a protein during translation. Here's what they all do in a nutshell. And so what we'll show you next is all of these guys in action. All right, so how do these molecules work together in order to make a protein? Here we go. So this is what all this shows is the cytoplasm of a cell. So if it's a healthy cell, it should have some free-floating amino acids in there that it's acquired through metabolism. And it should have, if it's a healthy cell, a bunch of free-floating transfer RNAs and release factors as well. We mentioned that the thing that's needed in order to make a protein, one of the things that's really needed, is the instruction booklet. Right? The messenger RNA carries the instructions on how to make the protein. It turns out that in order for translation to happen, the ribosome first has to bind to this messenger RNA. And it turns out that there's a sequence on the messenger RNA that kind of directs the ribosome where to bind. So we'll highlight that sequence here. So the sequence that tells the ribosome where to bind, it's called the ribosome binding site. And that's where actually the small subunit of the ribosome first binds. And the reason why the small subunit can bind there is because, remember we said that the ribosome itself is partially made of RNA. So there's an RNA sequence on the ribosome that's complementary to the ribosome binding site sequence on the messenger RNA. So because they're complementary, hydrogen bonds can form, and that's what allows the small subunit of the ribosome to attach to the messenger RNA. So in words, here we'll jot down what the ribosome binding site does. It's a sequence found on the messenger RNA that tells the ribosome where to bind to the messenger RNA. All right, we mentioned, so this is important, right? This sequence here, highlighted in yellow, tells the ribosome, the small subunit of the ribosome, where to first bind. Another sequence that plays a big role in beginning the process of translation is the start codon, and so here it is, AUG. And so basically what the start codon does is that it tells a cell where to actually begin constructing the protein. Right, so it's at the start codon where the first amino acid is brought into the process. So one of these amino acids is going to be brought to the start codon. The thing that brings the amino acid to the start codon is a transfer RNA. So it turns out that there's a transfer RNA that works specifically with each particular codon. The one that works with this codon is the transfer RNA that has the complementary sequence. Right, so this transfer RNA, UAC is complementary to AUG, so that tells you that this is a transfer RNA that's going to end up bringing an amino acid to this start codon. And so that's what happens. UAC, it turns out, interacts with the amino acid methionine, and it brings it then to the ribosome and the messenger RNA. And again, the reason why this transfer RNA can bind to this codon is because it has a complementary sequence. UAC is complementary to AUG. Once this first transfer RNA has brought the first amino acid to the messenger RNA, what happens next is that the rest of the ribosome, the large subunit of the ribosome, ends up clamping down on the whole process. And so now we're kind of seeing why ribosomes are needed during translation. Basically, the job of the ribosome is that it helps stabilize the interaction between this transfer RNA and this messenger RNA. Right? So it makes sure that the amino acid truly does get to the codon on the messenger RNA. And so what happens next is basically the same thing, right? So whatever the CCA codon encodes for, there's going to be a transfer RNA that brings a specific amino acid to it. So now we have the beginnings of a protein. We have the first two amino acids now strung together. You might remember that the bond that holds together two amino acids is called a peptide bond. And you might also remember that most bonds require some kind of enzyme to catalyze its formation.